Welcome to the TOBI Shark Tank. Please welcome your host, Dr. Steven Sampson.
All right, thank you. We are doubling down on the TOBI games. Family Feud seemed to survive. So this is unique Shark Tank Challenge. And so first I wanted to introduce you to a really important organization we partnered with. And again, TOBI's reaching out to other organizations around the world to support our efforts, the On Foundation. Their vision, On, is an international network and foundation focused on orthopedic tissue regeneration. Much like us, they believe collaboration is key. They have multiple international society medical conference affiliations and partnerships they support. They develop young talents, up-and-comers, through grants, scholarships, fellowships. Congratulations to our On TOBI Foundation scholarships from around the world. They also have grants, other On Awards, and pilot projects. So I encourage you all to visit their booth at the expo. They're a non-for-profit organization really invested in supporting the field. Here's their QR code. You can read about them, and you'll hear more and more in the future. So we're going to introduce our first speaker, and the way this is going to work, again, it's very interactive, where we have 3 finalists from around the world that had wanted to participate in this. And each will have 5 minutes to provide their presentation and pitch to the Sharks. We're going to bring our Sharks on stage. We have Kay Horsch from the On Foundation. Kay, if you want to make your way up here. And then we have Dr. Arnold Kaplan, who's going to be joining us, a.k.a. the infamous Godfather. Doesn't like the name, though. And then we have Scott Bruder as well. So if you can all make your way on stage and take these seats over here. And we will be the Sharks. And the contestants will give them a warm welcome. They'll have 5 minutes. So our first speaker is going to be Luca Ambrosio, and he's going to present on microfermented adipose stromal vascular fraction for degenerative meniscus tears in knee OA.
First up in the tank, this aspiring physician scientist hails from Rome, Italy, and has a particular interest towards translational medicine, with a focus on regenerative techniques to treat musculoskeletal degenerative diseases.
And then a buzzer sounds.
So, hi everyone, and hi to the Sharks. So I hope you're hungry, because I have some nice, fleshy, and fatty meat for you. So let's take a quick step back. So we know from guidelines that actually conservative treatments and surgical treatments may work well for mild and severe OA respectively, but actually we have a huge knowledge gap in between. So when treating moderate patients with moderate OA not responding to conservative treatments but not being at the same time candidate for surgery, that's where biological treatments are currently fitting in. And that's where our knowledge gap occurs. So we know now that fat is one of the most widely utilized sources for biological treatments. It's easily accessible, it's widely available, and it contains a lot of biological cues that have been linked to cartilage regeneration, anabolic function, and immunomodulation. And these are all packed in stromal vascular fraction. So we have some evidence about the effectiveness of SVF in knee OA and meniscal tears in both preclinical and early clinical studies. However, SVF isolation usually requires costly techniques that importantly limit the accessibility of care of regenerative treatments in most healthcare, public healthcare contexts. So that's why there is an overall lack of evidence about these treatments due to both the absence of large randomized trials and also the high variability among the utilized techniques. So I'm here to illustrate a technique that has been developed and patented from the collaboration between us and our plastic surgeons at our institutions that has been called stem graft. Actually, we perform a standard To measure liposuction, we need about 50 ml of lipoaspirate. We spin it at 1,200 Gs for 3 minutes, and we get this situation. We discard the oil and the infranatant, and actually what we get, we mix it for 50 times energically between 2 syringes with a cockpit, thus obtaining what we call the modified nanofat graft. So we save 4 ml of this graft, and then the remainder is spinned again using the same parameters. What we get in the end is this pellet, which is really, really dense in matrix and with a high concentration of adipose stem cells. The final injection is usually made of 1 ml of stem graft and 4 ml of modified nanofat graft. So we performed a kind of biological characterization study, and you can find here our graft compared to the raw lipoaspirate and another widely commercially available mechanofragmentation graft. So actually you can see here that there is a lot of matrix. So the laser is not, maybe is running out of battery, but you can see a lot of more matrix, a lot of more replicating cells, and a lot of cells expressing hematopoietic line markers, as well as the reduction of interleukin-1 beta gene and an increased expression of stemness-related genes compared to the other grafts. So what are we looking for here? We want to compare the efficacy of arthroscopic partial meniscectomy with subsequent SWF injection with this technique compared to meniscectomy alone in patients with moderate knee OA and concomitant degenerative meniscal tear who have not responded to conservative treatments. That's exactly where our knowledge gaps occurs. So we want to perform a 2-arm parallel group phase 2 double-blind RCT with a simple size of 30 participants as per our power analysis. We also accounted for possible dropouts. We will perform a 1-to-1 computer-generated simple randomization, and we will be, of course, double-blind. So our primary outcomes will be safety and efficacy, and we will see if patients will present any adverse events, and we will assess patients based on WOMAC and BAS scales, actually at 2 weeks, 1 month, 3 months, 6 months, and at the end of follow-up, we want to perform a knee MRI to assess for any structural changes. So why should you fund this? It's relevant. Just to use a trendy term, OA is a real pandemic. We need new treatments. It's safe. You can do it. It's autologous. You can do it right at the bench side in your OR. It's innovative. It's a new treatment. It's feasible. We are a large academic institutions. We see a lot of patients with this problem. We perform more than 20 arthroscopies per week. We wouldn't have any problems in recruiting patients and following them up. And it's cost effective because if you exclude the cost for the centrifuge and sterilizing on the liposuction cannula, the syringes and the needle are only $20. So you can do it whenever you want, whenever in the world, at no cost.
Great.
Thank you.
So we have time for one question you can stay up here. We're going to have one question for you on the hot seat. And then I also failed to clarify, the winner of this contest will receive a $10,000 grant from the On Foundation to support this research. So there's a lot on the line for them. And thank you for that. So do any of the sharks have a question for him?
What do you think the active component is in the material that you're injecting?
Excuse me?
What do you think the active component is in the material that will be injected?
Actually, what we saw in our biological characterization model was that compared to the other products, we had a lot of cells from the hematopoietic cell line. And, you know, apart from this, in this kind of derivative, we have a denser matrix that we think will also support the biological activity of this graft from a structural standpoint. So these two are the main differences that we noted comparing it with other products already in the market.
Great. Well, we're ready for our next contestant.
Thank you.
Next up in the tank, this Harvard University doctor believes she's developed an innovative and non-invasive treatment for cartilage defects that have great potential to restore patients' daily activity.
right, cool. So hi, sharks. So today I'm going to present one innovative approach for cartilage repair using a genus hydrogel. So first of all, I would like to convince you why we should invest on this new approach. So here comes a story of Roger Federer, one of the most famous tennis players in history, who has claimed 20 Grand Slams. Unfortunately, in 2016, Roger sustained a cartilage damage and had to withdraw from the competition through the whole year. Since then, he has suffered 3 surgeries, but still struggled for fluency. So while he's going through long-term rehab, he also has to face the fact that his knee might never be the same as before. So whether he can still go back to the court for another final still remains a question. So in general, people think, oh, cartilage damage only happens to athletes who are going through intense activities every day, but that is totally wrong. So cartilage is just a thin layer tissue lining around the bony surface of our joints, allowing us to move our joints smoothly, but it is so easy to be damaged that it can affect any one of us. So a sudden fall, like a wrong twist, or even repeated daily wear and tear can all result in cartilage injuries, leading to inflammation, pain, stiffness, as well as the frustration of not being able to get back to normal activity as usual. So, every year in the United States only, cartilage damage affects 1 million Americans, leading to 20,000 surgical procedures, 50,000 joint replacement, as well as a huge medical cost up to $850 million per year. Still, the complaints about current treatments has never stopped. So intraarterial injection of corticosteroids or viscosupplementation only provides symptom relief, so it never stops the disease progression. So eventually, most of the patients will have to go to surgery to repair their joints and restore the mobility. However, these surgeries are so invasive that it takes at least 8 weeks up to 2 years of non-weight-bearing recovery time. Still after that, more than 50% of the patients can never go back to their pre-injury level activities. So, the huge unmet need here is really about a non-invasive approach that can bypass the surgery at the early stage to repair the cartilage defects as well as restore the joint functions. We believe an ideal approach can be simple as a hydrogel injectable that will be simple to be injected to the cartilage, stay in the cartilage defect sites, allowing localized drug delivery as well as provide lubrication and mechanical support. So here what we are proposing is a genus hydrogel that satisfies all the above requirements. So it will contain drug-loaded tissue adhesive layer as well as a low-friction lubricating layer, which can be simply administered to the joint with a 2-step simple injection. So the first layer of the hydrogel will adhere to the cartilage defect site. It will also allow us to easily load disease-modifying drugs to promote the cartilage repair. The second layer of the hydrogel will be low friction and also tough, so it can provide mechanical support and also lubrication similar as normal cartilage does. So this unique design of the hydrogel will allow us to achieve unidirectional diffusion of the loaded drugs towards the cartilage defect site, and also allow the patients to resume physical activity after the treatment. So with this grant, I assembled a team of mentors from Brigham Women's Hospital and Harvard Medical School with synergized expertise in rheumatology, biomaterial design, intraarterial delivery, as well as regenerative medicine for translational research. So with this, we aim to achieve making the hydrogel and also evaluate their in vitro and ex vivo performance within 1 year. So the data we got here will be essential for us to secure future fundings to further push the project towards clinical translation. So with that, I strongly believe the success of the genus hydrogel will have the potential to revolutionize the future injectable treatments. With that, I will be happy to take any questions. Thank you.
We have time for one question.
Wonderful presentation.
Thank you.
I needed to ask the question because you promised to help our national hero, Roger Federer, to win again. And as you know, he's old already, so speed counts. So one question is, how far are you in the testing of the material? How about biocompatibility? And the next one, what will be your next steps if it works in vitro?
So the materials we're using here are all biocompatible. Think about like the origin that has been used a lot for like biomaterials, for like adhesive. The people have already been testing in clinical trials. So we are also we're able to control the biodegradability over time by varying the cross-linking density of the materials we're using. So that will not be a problem. So sorry, what's your next question?
Next question would be the next steps after your in vitro experiments.
Because this is like a proposal, and based on the expertise we have in the lab and also the collaboration with the MD doctors we have in the hospital area, once we have been able to show like the ex vivo like performance with low friction and also support like the mechanical loading, so then we will be able to move into in vivo studies to test like the drug release profile in vivo, like with the in vivo models, starting with small animal models and then moving into large animal models from there. Yeah. Thank you.
Last in the tank, this principal investigator from Palermo, Italy, aims to develop innovative technologies based on tissue engineering in an effort to treat musculoskeletal disorders.
Good afternoon, everybody. The main issue in the tissue engineering medicine is that cartilage does not regenerate. This is a big issue, and this is of big importance if you want to take into account that more than 250 million people are affected by osteoarthritis worldwide. And it is also important to take into account that osteoarthritis damaged cartilage and contribute for more than $136 billion on the healthcare cost. So today I will present you our now innovative biomaterial to treat osteoarthritis and to promote the regeneration of the cartilage. Today, the more effective treatment for osteoarthritis is the MACA, which is the matrix-induced autologous chondrocyte implantation. Very quickly, in the first step, we have an autologous harvesting from a non-bearing zone of chondrocytes, which are then expanded in vitro and reimplanted after assisted by a 3D matrix in a scaffold. Then the scaffold is reimplanted in vivo to promote the regeneration of cartilage. But the step 5, the most important, is the rehabilitation. Why is important rehabilitation? Because it promotes the production of type 2 collagen in the cartilage, so hyaline cartilage. But there is a big but. It's a long process, and there is a low patient acceptance. And there is no consensus today about optimal therapy for each patient. Our material is included in this complex scenario and in the step 3 to promote the rehabilitation. How? To improve the effect of the rehabilitation and to improve the compliance of the patients. But how does it work out, our biomaterial, and why it is so innovative? Our biomaterial is composed by piezoelectric polymer embedded with magnetic microparticles and cellularized with chondrocytes or progenitor chondrocytes harvested during a MACA procedure. Importantly, our biomaterial is responding to an external pulsatile magnetic field which induce cyclical contraction and expansion of the biomaterial. In this case, we have a double effect. The first one is the electric discharges from the piezoelectric component of the polymer, which contribute to stabilize a chondrocyte phenotype towards the production of type 2 collagen. Most importantly, the contraction of the material induce a mechanical stimulation directly at cellular level. So we can have a big contraction, a perfect contraction on the cells embedded in. And this is very important because by modulating the magnetic field, the external magnetic field, we can go and counter the patient's response. So we can promote a tailored rehabilitation on the patient, directly on the patient, by modifying the external magnetic field. We already produced a a prototype of this biomaterial, which, as you can see, at time 0 had like 2.3 centimeter diameter. This is an RX. After, we'll go faster. After MRI, we find that the material contracted up to 1.9 centimeter, as you can see in the sagittal and in the coronal view in the MRI. And interestingly, after 6 minutes, so after 2 minutes of recovery, we found that the material expanded again up to the initial dimension. But what happened at the microscopic scale? As you can see, microparticles embedded in the material moves when we apply an external magnetic field and also contract the material. This is a different view, a big scale. Okay, sorry. So to summarize, we already fabricated a prototype of this magnetic responsive scaffold, and this scaffold responds to an MRI cycle, and there was a contraction, a reversible contraction up to 50.2%. We plan to use it with external coil or with external. I want to say thank you to my team, medical doctor, orthopedic surgery team, and Professor Riccardo Gottard in the Penn University.
Great. We have time for one quick question. Dr. Bruder.
Thank you very much. It's a fascinating technology trying to address a very large unmet need. Can you explain how your technology supports the adhesion of the eventual construct if it's continuously getting larger, getting smaller, getting larger, getting smaller? How do you eventually get to the place where it is adherent to the native cartilage underneath or the subchondral bone?
I mean, we can program the expansion, the contraction of the material just inside the articulation after implantation. And by providing a different intensity of magnetic field, we can check how much the material contracts and expand itself by following by MRI, and to see if it is enough, it's not enough, to follow the patient. And the patient will come back, but it will be happy to come back because he do not have to do any movement or any something like that.
So the money is for clinical study or basic science work still?
I can't hear well.
The money is for a basic science or clinical study?
No, now is not clinical study, but we are ready to start with clinical study because MACA is already approved. The material we use is already approved. We are the equipe already ready to start with clinical studies in Bucca della Ferla Clinic. So we are almost approaching to clinical studies. Thank you.
Great. Well, we've heard from the contestants, and Jeff, you want to put something up in terms of a vote for the audience, and then each of the sharks will make their vote as well. So you can see on the top board we have our first speaker, our second speaker, and our third. And again, their votes depend on you for $10,000 for their research to support. And so we'll see where the audience pans, and while that's being calculated, Kay, do you have a vote for one of the three contestants?
I'm most fascinated by the last approach, the smart biomaterials. I would bet on that one.
I'm going to support the smart biomaterials as well. I think about it from a product development standpoint and what can be achievable.
Yeah, I would vote for the first speaker.
Because it has cells.
Of course, I'm a cell guy.
And I'm personally really excited about the use of machine-based mechanical energy to stimulate our body's own endogenous healing in cells. So I would vote for the third speaker using the piezoelectric energy to stimulate cells. And the audience.
And we have a winner.
All right. Do we want to bring the winner out and congratulate them? Is our winner. Bring him out. Congratulations. Way to go. Congratulations. Well, thank you, everybody. Congratulations. Thank you, Sharks, for our first TOBI Shark Tank.